Pardon, more technical.
We really rely on our volunteers. Sorry, y'all. I forgot to share.
Ritchie. All right. Good evening. Happy Earth Day. All right, so if you haven't met me before, my name is Ritchie. I am the conservation manager at the San Antonio Botanical Garden. I do a lot of field work, and I grow a lot of plants. I grow a lot of weird native plants from all over the state, and it's a lot of fun. So this was a really interesting project that I got to get involved with after chatting with the folks here, and I'm glad I did this and also frustrated I did this. This is a really interesting project to take on, and it was quite an endeavor for a grower to keep track of some of this stuff. So without further ado, let me give you a bit of an introduction, right? So cotyledons, what we refer to as the starter leaves of plants, we also often call them starter leaves. They're not really leaves. They're actually part of the embryo. They're part of the embryonic progression of these plants. So they're not considered true. They're the embryonic leaf in seed-bearing plants. That's an important distinction, right? Your bryophytes, your lycophytes don't do this. This is something that is unique to seed-bearing plants, you know, not just vascular plants in the traditional sense, the way we think about them. And they're the first bits of tissue that form after germination. And I mean immediately after germination. Some are already formed inside the seed, and some of them will form more fully as the seed emerges, and as soon as it hits oxygen, it's like, all right, let's go. So the purpose is to, you know, prep that seed for its next stage of life. Now, this is a really frustrating thing to document. It's a very frustrating thing to document. They're very fleeting. This represents the shortest stage in life, and they're incredibly difficult to photograph because they're so small. They're not large, you know, big showy leaves that you can get a lot of light into. Like you can see, like this one is casting a shadow. That is probably the size of, you know, my pinky thumbnail, or sorry, pinky nail. But like it's very, very small, and you can see like I was barely able to capture the trichomes that are there, you know, on this. And so what we're looking at right now is this is the cotyledons are the larger leaves, and those little bits next to it, those are the true leaves barely starting to emerge. So they're very fleeting. You have to be at the right place at the right time. You blink and you miss them. Some are more broad, like they are broadly applicable to certain family or genera, and some are they're much more very like specific based on the species. And there is no real hard and fast rules. They have a lot of morphological differences, same as everything else. But the disclaimer here is that I'm not a paleobotanist, first of all. I am a conservation botanist. And so I got as much in-depth information as I could give you guys. And I'm also not a professional photographer. So give me some grace. It wouldn't be a presentation of mine without a cladogram. I'm going to try and actually move. We're going to move this a little bit just so you can see kind of what we're dealing with. We have very early stages of life here represented at the bottom. And as things get wider, you can see how, you know, we're starting to get those embryophytes, the tracheophytes, and finally, you know, you get to this kind of seed-bearing world. And this is kind of where this starts, right? Up until this point in the evolutionary tree, things were very simplistic. They were very slow to grow. Things had very simplistic cell replication. This is really basic life forms. And for the purpose of this, we're going to keep things simple and practical. We'll stick to smaller, more common groups, right? Things that we know today, we can recognize today. And these are relatively small taxonomic sections when you're looking at the wider tree of life. And they actually represent an enormous number of plants. There's a ton of biodiversity in just these categories, because this is kind of where plants that we know today, modern plants, really began to branch out. So, what's our timeline look like here? All right, in the beginning, 1.5 billion years ago, that was a long time, we have aquatic plants. We have the basic building blocks for plant life coming from the ocean. And it's not until around 470 million years ago in the Ordovician that we finally get evidence of land plants, right? This is that really early stuff that we were looking at the cladogram a second ago. When we get to the Silurian, 443, emergence of bryophytes, we're still not at seed-bearing plants. 425 million years, the late Silurian, we finally get vascular plants. These are like true vascular plants the way we understand them. These are the early ancestors of some modern day ferns and some weird things that are kind of holdovers from ancient periods. And then around the Triassic, 251 million years ago, we finally get seed-bearing plants. They are the dominant. I mean, sure, the evolution tree is a little weird here, but we do get the dominant life form on the planet being a seed-bearing plant. This is the age of conifers. People often refer to the world being covered in redwoods. And this is the period that we're looking at. The Triassic is really where these plants become dominant. And then we finally get to the Cretaceous when their angiosperms become dominant. So this is the most recent evolutionary period around 140 million years ago. And at this, I mean, this is the time scale of the planet Earth, it's really not that long. It's a pretty short section. And we finally get the early adaptations of these beautiful flowering plants that we all know and love. So it gets weird. Gymnosperms, right? So here's a couple of classic examples. I've got some photos of an absolutely massive Douglas fir that's not very common in Texas. There is a population kind of that exists in West Texas, but this is in Arizona. This is in the Chiricahua Mountains, and you really get to see some stunning conifers there. And to our right, we have one of the most famous gymnosperms, Ginkgo biloba, and that one is an oddity in itself and definitely a relic of the past. So gymnosperms definitely fall into the category of cotyledonous plants. There are no absolutes, right? In this group, things get a little weird. The lineages are a little bit different, and they don't really resemble angiosperms in early stages of life. But there are some really interesting things between their life cycle because they have 2 to 18 cotyledons. Remember, these are the first seed-bearing plants. This is the genetic blueprint for what we have everywhere. This is modern day blueprints here. Pines germinating is pretty crazy to watch. You can almost watch it in real time. And some species in this group do keep their cotyledons under the soil, and some, will present them as normal, like some of the others that we're about to look at. So here's a couple examples. I have 2 gymnosperm families for you, the Cuprosaceae and Pinaceae. Does anyone want to take a guess as to what genera we have presented? No wrong answers.
Aloe.
What's that?
Aloe.
No, so not aloe. These are, I'll give you a hint. These are both conifers. There is a pine on the right. It is not, well, I mean, some people call it cypress on the left, but that's a red juniper, Juniperus pinchottii. And on the right, we have the Texas famous, that's the Pinus remota. That's our one true pinyon pine here in Texas. And that collection was in West Texas. So we have a juniper and then we have a pine, and you can see that little cage that's around the leaves on the pine that's still attached to the seed. Those are the cotyledons. Now the juniper cotyledons are those really long stretches of leaves on either side. Juniper only has 2, but the pine, I don't know, it looks like maybe 10 to me. I didn't count when I took this photo. I was lucky enough to have this from a holdover from last year. But we have a lot of pines that germinate like that. It will push the seed actually out of the soil, and the cotyledons will actually start to become chlorotic before they actually open up, and the true leaves will push the cotyledons open. It's really crazy to watch. This is one of those where you can almost watch it in real time. You just remove the hat, you watch it unfold. Okay, so let's go to another kind of weird branch on the evolutionary tree. These use some very bizarre pollination strategies, right? So on the left, the photo is the famous magnolia, grandiflora, and on the right, we have a West Texas Aristolochia coriae. And magnolias are probably the oldest relics we have of the first angiosperms. Definitely, I mean, magnolias, for sure, are a very bizarre relic species with an ancient lineage. The current representatives definitely vary widely on the evolutionary scale in size and in flower presentation. But definitely, like I said, the modern day Magnolia grandiflora is a relic, while Aristolochia kind of represents a more recent adaptation. Being the earliest flowering plants, surprisingly, they have the lowest percentage among the angiosperms, only about 2%. Despite that, they are very well known. And a lot of them have a lot of medicinal value as well. And if you've ever watched a magnolia in full bloom, it's full of beetles. It's the last thing you would expect. And those flowers are kind of like bones. So the pollination strategy is definitely different from traditional flowers. And Aristolochia has a very foul smell. It's one of those rotting meat smelling flowers, similar to Amorphophallus, the corpse flower. I don't have any photos of these. This is where I have to apologize. I have grown a lot of these magnolias, but just for the purpose of like the 4 months I had to do this like pre-study going into like a longer study that we're gonna do, there's just not an opportunity to photograph this really interesting group of plants. It's so narrow, and we've grown a ton of them, but I didn't have any cotyledons to show you, sorry. So we'll move on to the real reason we're all here, the monocotyledonous angiosperms. This is where we start getting into some weird kind of confusion, resulting in like people arguing over paleobotany and lineage and things like that. And so these are monocots, and monocots vary widely across the evolutionary scale and just on a morphological scale. Right, monocots not only include yucca, but they also have Liliaceae, it's represented in there, and you also have grasses. Grasses are monocots. There's a lot of weirdness and a lot of overlap between pollination strategy with the dicotyledonous plants. So they cover just as much ground across the taxonomic chart, and they're regularly associated with stuff that I mentioned like grasses, Asparagaceae, but you can also make the argument that they're a more specialized version of dicotyledonous plants. It's believed that the monocots came after the dicots, so this is kind of a more focused way to grow and present their strategy. Like this is a newer evolutionary trait. Not quite as many species, right? Only 23% representation in the angiosperms. But these have evolved, not long after dicots, and they do use the same basic pollination strategies. You can expect to see the same pollinators visit a lot of these plants as others. There are some exceptions, yucca, the moth and bat pollination strategy is a little bit more narrow, it's a bit more niche. So what do these look like? Do you like my super out of focus Poaceae representation there? Yeah, I'm telling you, these things are really hard to photograph. In the center, we have a commelina, right? That's our beautiful blue dayflower. I'm assuming that's what that was. It's a weed all over the place in the grow area, so I'm used to seeing it pop up. One of the reasons I didn't include a lot of species names was because there's a lot of mystery here still. And I wanted to kind of keep everybody guessing, because when we get later on to some of the dicots, you're going to be confused. The Asparagaceae we have represented it's actually, that's my one exception to the rule. That is not a Texas native. It was the only Asparagaceae I had the opportunity to photograph during the course of these last few months. And that one was collected I think that was also collected in Chiricahuas. It's a Triteleopsis. It's a beautiful flower. I hope to bring some to one of the NPSOT meetings in the future, because I imagine we're going to have just oodles of that. Monocots are insanely good at germinating. I have almost 100% germination with monocots. A really interesting observation of their strategies too. But you can see there's a lot of variability, right? These are all different families, so you would assume to see some differences, but there are no real kind of unifying features. Each one of these plants is presenting itself differently, and at least in the center of the cotyledon, you can see that larger leaf, that's the original leaf. That's the original cotyledon that emerges, and that little curled leaf coming off the side, that's the new growth. And it looks completely different from the closest, comparison we have to the Poaceae family. And even Poaceae, you can't pin those down. It's different between genera. So here we go. We're going to call these eudicotyledons because we had to kind of reform dicot. Dicot does not really fit the overarching structure, right? Because we can't just refer to them as dicots because they're not all true dicots. Di meaning two, because the gymnosperms are eudicotyledonous and they have 18 cotyledons at times. So dicot was not an appropriate naming strategy for these. So I have a mint here photographed at Enchanted Rock. You guys should definitely go with Jerry if you haven't been. Wow, what a place. And then to the right, that's Arctostaphylos pungens. And we actually do have a population of that in Texas. We're lucky to have some in the garden. Beautiful, beautiful tree if you've never seen it before. All the Ericaceae, the blueberry family, are just absolutely stunning. So this is by far the greatest representation of flowering plants, right? This is the most traditional, straightforward, you can recognize them pretty easily. These plants used to be grouped into dicots. Genetic research led to a new category. Basically, it came before dicots. Not all dicots can be considered eudicots. It's one of those things. It's like, hey, not all cacti are succulents, but or not all succulents are cacti, but all cacti are succulents. This leaves a small percentage of ancient plants without an easy explanation outside of the cladogram, which I will not subject you to. I just wouldn't wish that on anybody. For a modern application, eudicot is what we're going to stick with. And 75%, I mean, that's pretty much everything when you think about it. If you encounter a flowering plant in the wild, chances are good it's eudicotyledonous. So here's some cool families. Thanks for joining, Judy Osborne. I'm going to close this window real quick so you can read that family name.
Top two while you're at it.
Okay, yeah. Zoom is fun, isn't it? Hey, we make it work. In this world, take what you can get. So I'm gonna burn through a bunch of families real quick, and I'm gonna subject you to total chaos because there is no unifying strategy across any of these families. The first one, Apiaceae, that's a weed that I photographed in the garden, in the rare and endangered garden at Selah. And this is one of those, I couldn't really get the species down, but it's one of those wild carrots that we all know and love with those really cool like branching leaves. The one in the middle from the family Pixaceae, this is one of my favorite plants that you can find in the hill country and a lot in South Texas. It is Amarexia rhyea. We love that plant. Beautiful bloom, very, very bizarre growth strategy. You can see like the cotyledons are really large, the leaves are much larger and deeply lobed. So even in plants that have deeply lobed leaves, you still have this very smooth, round, uniform cotyledon. And you can kind of see the lobes emerging from that new foliage. Those are the true leaves coming up there in the middle. But the Pixaceae is very neat because you can see kind of this fan presentation of venation on the cotyledons, as opposed to like Apiaceae, you barely even see that central vein that's running through the middle of the cotyledons. And they're very long and narrow, and kind of similar to the juniper we were looking at earlier. This Calicantha, occidentalis, that's a really interesting one. It's got a really weird range, but it kind of has this, offset, like rhizome looking weird cotyledon where it's, a rhombus almost, but it has this like weird curl right there at the base of the stem, like just a weird cotyledon. But it seems to be symmetrical, which I thought was really interesting. Those points are, oblong in the same direction. And that was a really cool one to photograph, and I was really lucky to grow that plant. Michael collected that and brought it back to the garden. Calicanthiaceae, that's a weird family. Interesting distribution too across North America, worth looking at. So the Malvaceae, that's going to be much more recognizable, right? It almost has that mallow leaf. The cotyledons almost look like the true leaves. That's kind of like five-pointed, like five-sided leaf that we're used to seeing, that pentagonal shape. And this was a wild plant. This is another species I couldn't really pin down. I'm pretty sure what the genus is, but I don't want to put any misinformation out there. But it is 100% sure a mallow. And I wish, probably like Turk's cap will be recognizable to a lot of you. You probably have seen it germinating in your gardens when you plant, like Turk's cap reseeds a lot. If you're lucky enough to catch it, you'll be able to see like definitely that pentagonal shape will present. And I really like this one because it has 3 very strong veins to kind of frame that leaf when it comes out. And then the 2 next to it are euphorbias. These are both weeds. These are just things that came up in pots in the garden. So the one on the left is I'm 90% sure that this is that green poinsettia that we see everywhere. And every single one of the starter leaves I saw had that weird misshapen appearance. So at first I thought, hey, maybe this is, you know, some weird thing that happened when it was germinating. It wasn't able to escape the seed. It received some damage on the way out, and it was symmetrical. So I figured, okay, well, you know, this is odd. But no, every single one of these that I photographed had that same morphological shape. It has this weird bulbous appearance, and then this very clean swoop right after that dip. And they each had that like melted spatula kind of look to them. Whereas the one to the right, that's a spurge. You'll all recognize that as that, you know, nice red purple leaf spurge, the very small leaves that we see emerge in anything. If you're growing plants, this plant is the bane of your existence. It will come up in everything, everywhere. The seeds are tiny, they explosively dehiss, they're just a lot of fun. These cotyledons are probably the smallest, definitely the smallest that I photographed. I was lucky to get such a good photo. You can really see the venation and that plant. Very, 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 very small. But they don't present any color in the beginning. And, you know, obviously, like when they grow up, there are all kinds of different shades of red to purple. But every single one, every cotyledon that I saw, every single one was green. That same color green. Okay, so I'd be surprised if people can't recognize the Canabaceae. Anyone want to take a guess? Again, no wrong answers. All right. It's a tree. Yes, it's a hackberry. Nice. Always recognize a hackberry because it has that indented end, like that point always comes. You've got 2 points there at the end of the hackberry. I mean, they're everywhere. You'll see this, you'll never be able to unsee it. And it's cool, like, and this is unique. Because you can definitely tell, like, this is different from other plants in the family Canabaceae. This is very unique to the genus Celtis. Not like anything else, but very recognizable. Anytime you see this cotyledon pop up in one of your potted plants, it's time to rip it out. Time to rip it out. The middle one, the Gentianaceae, that one is actually that's a really cool eustoma that we collect in the hill country. Exaltina is a species. And I really liked that the veins barely presented in the middle. It almost looks like it's been pressed down, like flat, or plumped up to so the veins don't express themselves. And all you could really see from the venation was that very, you know, very subtle indentation there in the center. And I loved that it came to a point. That was something I didn't see in any of the other plants that I photographed. At least like that very round and very distinct tapered end. Very unique, very cool to see. And by the way, if you've never seen eustoma, oh man, it's a stunning, absolutely stunning plant. And the one next to it in the Polemoniaceae, anyone want to take a guess? I'm sure you've grown it before. It's a biannual. It grows for 2 years, flowers in the second year, then dies. Standing cypress, Epomopsis rubra. You can kind of see the leaves starting to present themselves there on the far right. They're starting to kind of pop out a little bit. But the cotyledons were very uniform, very neat, very tidy. And I mean, this plant just germinates like crazy. You can't stop it from growing. Let's go on. Okay, very interesting from the Rosaceae, very interesting kind of presentation from them. I'll give you a hint. Both of these plants are in the same genus. I was able to photograph two things in Rosaceae. They're from the same genus. They're flowering. I mean, they're really lovely fruit bearing trees. That's what they're famous for. So these are both wild collected prunus. The one on the left, Prunus serratina, that's our escarpment black cherry. The one on the right, Prunus minutiflora, that one was collected in the Blackland Prairie outside Wilco. Have Native Plant Rescue to thank for those. So these are really bizarre because these are true leaves. These are not cotyledons. This is one of the examples of cotyledons kind of emerging beneath the soil. If you're lucky and the seed is really close to the top of the soil, you might see one. But most of the time, the cotyledons are going to present beneath the soil surface. So what we're seeing is the emergence of these weird, misshapen true leaves that are just struggling to get a foothold. The Prunus serratina germinated in a much more uniform fashion, but the minutiflora was they just took whatever they could get. They were wild, a really wild plant to photograph. Okay, I know you guys know the Solanaceae, the one on the right. It's everyone in this room is going to be like, oh. They're tomatoes. They're tomatoes. I went next door to the greenhouse where they grow the vegetables. And they're like, oh, there's some tomatoes. I gotta photograph those. But yeah, I mean, you can see they're very neat. They're very tidy. They're very happy. You know, they're not going to last long. I mean, maybe a day before they have true leaves and they're taking over the place. But those were all uniform. Every single one of them looked exactly the same. Very, very cool. All right, I've got two really cool ones from Plantagenaceae. These are pretty recognizable plants. I'd say people in this room have definitely pulled over the side of the road to look at these blooms. Any guesses from Plantagenaceae? No, bluebonnet's a legume. So these are going to be, I'm trying to think, I don't want to give it away. Okay, so you're driving down 281 and you see something beautiful. You're like, oh my God, what is that? And you pull over and it's this tubular bloom at the end of this long stalk. And they're not too tall, but they're just tall enough to spot above the incessantly mowed grass. What was that? Not paintbrush.
Yeah, that's
Instant, yes. See, you just gotta set a scene. And people are like, I pulled over for penstemon before. These are two species of penstemon. One is cobia, one is triflorus. Anyone want to take a guess which one is the triflorus? Ooh, it's the one on the left. The reason why I know it's triflorus is because it presents red. Triflorus has a much darker leaf, and the cotyledons have that same red streak down the middle. And you can kind of see the red beginning to present in the true leaves. But the triflorus always has that darker leaf. And if you pull over and look at the cobia, you're like, oh, it's kind of light, like it's light like the flowers. Everything about the triflorus is darker compared to the other penstemons. It's a real oddity in that genus. Now, the one on the right, I don't think anyone's going to have trouble recognizing. That's a cedar elm. I mean, that's your other tree weed that's popping up in your potted plants. This is one that I save as often as I possibly can. We use it for bonsai presentations on a regular basis. It's just the easiest tree in the world to work with. But every single elm I've ever seen has that very same cute little round, kind of curled on the edges, cotyledon, where you can't even see the venation. It just looks like this that green thing. But every single elm I've ever germinated looks exactly the same. I can't tell them apart until they're a little bit older. Okay, Apostanaceae. I'm gonna give you a hint. All of these plants are in the same genus. Every single one of them. If anyone knows their plant families, I mean, it's just it's gonna be a weird one. But these are all in the same genus. So this is a really good example, right, of the variability in these new starter leaves. So these are all Asclepiads. Every single one is in the Asclepiads genus. The one on the far left is Asperula. That was the weirdest one, I gotta be honest with you, because it curled on the ends, and each single one did this. The cotyledons did not last very long on this plant, maybe, you know, a week if they were lucky, and then they fell off. But they were all this nice lime green, very smooth, very even, and they almost had this like translucent appearance to them. Like you almost feel like you could see through the veins. The one in the middle is a Western milkweed. That one was, oh no, this was the Eastern milkweed, that's right. This is the Viridiflora. And so this one was collected, probably Fayette County. And then the one on the far right, that's the Angustifolia. So very different presentation all across the board. And what surprised me about the Asperula was that the cotyledons almost seemed fused. I didn't see how a stem could emerge from that. But in the center one, you can definitely see, yeah, how they branch and they split apart. And then, you know, as soon as they come up, you've got a big stalk. Really interesting plants, really fun to watch them grow. Very, very frustrating genus of plants to grow also. Very picky in cultivation. They will tell you as soon as they're unhappy. This was a really fun one for me. We have a lot of Styrax at the garden, and that's what we're seeing here. We're seeing two different subspecies of Styrax plantanifolians. On the right, we have subspecies Youngye. And on the left, this is subspecies Texanus. And these were very uniform. There was almost no differences. I think if you really wanted to split hairs, you might be able to count the veins to tell them apart. But I think you're best waiting 10 years to figure out what kind of plant you have. Sorry. But yeah, obviously, very little differences. These are both subspecies of the same species. I mean, there's very little variation here. But the you can kind of see in the background of the left-hand photo, those are some fresh cotyledons, very pale. And they didn't green up until they finally, you know, got some nutrients flushed in them and began to curl out. But each one curled in its own funny little way. And so it was really fun to watch them just kind of emerge and do their thing. All right, we're getting into the weeds here. Asteraceae. One of these is not native to Texas. One of them, I'll say, is an eastern species, and one is an unknown aster collected at Big Bend. Because we were sent the wrong plant. Someone told us they gave us some seeds, and we looked at them like, this is an aster seed. This is very clearly from an aster. So we don't actually know what we have yet. We're trying to figure it out. But it's from a very remote population in Big Bend. The one in the middle is not native. It's a zinnia. I wanted to include it because I did have a lot of photos of asters, but I felt like, of course, you know, the morphological differences in the family Asteraceae is vast, to say the least. So I think it would be cool if we had some representation of that also in the cotyledons. Very surprising differences though, like, you know, some of the other ones you've seen like some similarities, but this is just all over the place. You would not be able to pin down the family just by seeing these plants germinate. Maybe the one on the right. The one on the right is Monarda viridissima from East Texas. The Monardas, I think, are, you know, they let you know what they are. They're fuzzy as soon as they come out, and they express their color almost immediately. And it doesn't take long to get true leaves in the Monarda. And you can see there's kind of a different strategy. The one on the left, that's the unknown Big Bend plant. And that one, I think, kind of grew the slowest, but each one of these plants, as soon as they got true leaves started, they would be just as big as the cotyledons, and then boom, a second pair of true leaves, and then another pair of true leaves, until eventually they were just all of a sudden had these huge leaves that shaded out all the lower foliage. Really interesting growth strategy, and it was interesting to see that when they emerged, they all had a similar kind of approach to getting started in the early stages of life. Just as frustrating to identify cotyledon-wise as they are flower-wise. Yay, asters. Okay, some more well-known plants here. Full disclosure, the middle plant, I'm not 100% sure because it too closely resembled what I had growing in the pot. There was something else that kind of tipped me off about it. I will say the outer two photos, the left and far right, those are both salvias. The one on the left, Engelmannii, that was a really cool one because all the cotyledons presented the same way. The one on the right, Azuria, did not present the same way. They had some differences. Some more closely resembled the Engelmannii, some more closely resembled, I don't know, like a legume. But it was interesting with the asters is, with the exception of the center plant, they all kind of had this flat kind of start to them with a big arch, like a big, wide presenting arch. But they all were flat right there in the center. And this one in the center, man, that really stumped me. It was really bizarre seeing that because, oh, never mind, that's Salvia pensteminoides. So yeah, take a look at the penstemon. The one on the left is Penstemon copia, and then the one in the center, that's Salvia pensteminoides. Yeah, so even in this cotyledonous stage, it kind of resembles a penstemon. Really strange. Really raises a lot of questions. So we're gonna get to the weirdest stuff that I photographed this year. This is kind of your opportunity to look at oaks and say, you guys just do your own thing, don't you? This is another one where the cotyledons mostly present below the surface, but that middle photo, whew, that's a Quercus polymorpha that emerged above the surface. And a lot of people will plant their acorns above ground, and a lot of the ones I photographed were, but they didn't really do this. They would just immediately go into their true leaf form, which is what you're seeing at the top there. And even on the far left photo, those leaves at the top, those are not cotyledons. Those are true leaves. So you can kind of make out what could be a cotyledon there about, I don't know, a third of the way up that stem. And that's kind of what they appear as beneath the soil. It's the same thing you could expect to see in a ginkgo biloba, but all oaks present in this way. And I've noticed this trait all across this family. I've noticed it in walnuts, I've noticed it in pecans, all the hickories do this. And so it's a pretty unifying factor. As soon as I germinate some beech, I'll let you know. But the one in the middle is really interesting because you can actually see the embryonic tissue, right? That was inside the acorn, and it expanded. As soon as the seed emerged, those halves of the acorn expanded. They flushed with tissue. They became red. They had pigment to them, just like the new leaves flush red. And then just above that, you can kind of make out this very little baby branch. I got the mouse, so I'll point it out to you right there. This little baby leaf. And whether or not that's cotyledonous tissue or true leaves, like really a lot of people consider that those portions of the acorn that are already inside, that's the embryonic leaves. So we're seeing the expression of that. I mean, there's people say, oh, that little thing, up the stem, like that's a cotyledon. But to me, in oaks, that really strikes me as a cotyledon. And all the plants in this family will germinate that way, split open, and then that's where the stored energy is. But to see it stretch and emerge like that, pretty awesome, pretty awesome. I could bore you all day with oaks. I grew about, 12,000 of those in the last year, so not exaggerating. Okay, I'm gonna get on to my favorite family. This is my last group of photos I have for you. I love the legumes, always have, always will. And there is some uniformity, right? We can kind of see some similarities in size and shape and the presentation of veins, but there's definitely some exceptions here. And one of the most interesting things I found are between the two bottom photos. So when we looked at the photos from the styrax, the subspecies of styrax, there's almost no real visible distinction between the two. Maybe, but really not. These two bottom photos are plants in the same genus. Not just that, they're the same species. They're different varieties of the same species, wild varieties. So on the far right, that's Cercis canadensis variety mexicana, Mexican redbud. The one right there on the bottom left, that's variety texensis. So that's our Texas redbud, and it was really striking to me because Mexican redbud has a much glossier leaf, and it's undulate, which means it kind of has this wavy kind of margin to the leaf. And the cotyledons on the Mexican redbud kind of curl, almost like a curling leaf. And just like our Texas redbud, it had these broad, flat, matte leaves. The Texas redbud doesn't really reflect light. It kind of acts as this really broad, absorbent material. It's always very matte in presentation. It does not have that glossy finish that you're used to seeing in some Eastern redbuds. So I thought that was really interesting. And they don't really even have the same shape to them either. There's not a lot of uniformity between the two. And you can kind of make out, like, I think the bottom left one, it's very fabaceous. It makes you think fabaceous when you see it. I see a lot of Senna present that way. Both the Senna Locatii and the Lindheimer that we grew this year had cotyledons that resemble that. Pretty interesting. And then the one in the center there, center right up top, that is actually a Bauhinia lunarioides. That's the wild Anaconcha orchid, which, I mean, it's a bit of a misnomer because the Anaconcha orchid comes from the Anaconcha Mountains, where the orchids only bloom white, but most Bauhinias bloom pink. So this is actually the true pink wild collected version of that plant, that species, instead of the white cultivar. So you can kind of get a feel for what the Bauhinias look like. But again, it's kind of hard to see because the other one is tilted up. Again, they're really tough to photograph, folks. But you can kind of see that wide swoop there on the right side of that bottom leaf that we get a really good photo of. Again, like some of the previous cotyledons, they mirror each other. You know, it's like holding up a mirror. Pretty interesting. And then the one way up in the top left, you'll never guess. That is a mesquite. It's a screw-bean mesquite. So if you don't, I mean, a lot of fun, not fun, boring botanical facts about mesquite was that it used to be Prosopis, right? All the mesquites were lumped into Prosopis, and that genus was dissolved. And now they've been split into Naltuma and Strombocarpus. So this is the one that ended up in Strombocarpus. And I think it's pretty fitting because morphologically, it is quite different from, you know, the honey mesquites and some of the other ones that we see, you know, like South America. It's weird. It didn't look like a legume to me. I was very surprised. This is probably the one that surprised me the most. But I had a lot of fun. I took a lot of photos of legumes, but not all of them presented as well as these did. So these were my picks. So can you ID a plant by its cotyledons? Well, sure. I really wouldn't recommend it though. It's quite tedious. I don't think the juice is worth the squeeze here. As far as ID is concerned, the variations between a generic can make it difficult to pin down the family. I mean, forget the species. Plus, it is the shortest stage in the life cycle of all plants as we understand them. We can definitely learn from this process, but I wouldn't necessarily add it as a tool in your arsenal unless you're propagating on a regular basis. That's the exception, I think. Because if you are growing plants, this can be a helpful tool because you can distinguish the invaders. There are merits to that, and know what you're working with. You want to be able to identify these things quickly sometimes, especially if you lose a tag. God forbid, you know, you lose the tag for the one tag for the flat of plants that you have, and it just happens to be presenting in a very strange way. If you can recognize those cotyledons, it can really help put you in the right direction. Plus, you know, if you tell the differences between things like penstemon, you know, it can really help propagation-wise. You can know if you have some, you know, some cross-contamination in your flats. So there are merits to being able to tell them apart, the invaders from the desired material, but don't go chasing waterfalls. Stick to the fruit and flowers that you're used to. There are much easier ways to identify plants. We've been doing it for a long time. I would stay there. If you're interested in this, I would point you, there's some really handy articles at Science Direct about this. If you just want to do some reading, it's a rabbit hole. I'm telling you, the research is very in-depth. And most of the research on these cotyledons is being done by species. I mean, forget genus, forget family. They're talking about very, 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 very specific morphological traits. And really, that's just a tiny fraction of the research that's being done. So you can dig and dig and dig and dig and dig for hours, and you won't really learn that much about morphology. But there are some really interesting, cool things, and definitely like that's where my playground came from, and that's where you can find a lot of these research articles on paleobotany, which is really, really fascinating stuff. It's not the most exciting material to examine, but it is pretty cool. So I'll leave you with the best advice my friend Mac ever gave me: have fun and be yourself. Happy Earth Day, folks. Okay, I've been here enough times to know that this is the section I'm going to dread the most tonight. I'm going to open the floor to questions. Yes, sir.
You said that the monocots had a really high germination rate,
Yes.
and like bluebonnets, a third is considered very successful the following year. And that was dormancy to protect the plant. And you're gonna have to repeat my question in a condensed.
Oh, yes. I'll summarize.
And that's what I'm just trying to figure out. How did they survive evolutionary if they didn't delay dormancy?
This is a great question. And this is kind of tying into something else I talked about earlier. But the question was, because I mentioned that I had an insanely high germination rate with monocots, but a much lower germination rate with some dicotyledonous plants. And the example given was bluebonnets, how they have a dormancy period, takes a long time for them to emerge. So how do the monocots survive if they don't have this kind of delayed germination survival strategy that a lot of plants do? And the answer is, it is a numbers game. They're just flooding the world with as much seed as they can possibly put out. And that's really one of the most surprising things. Like, if you're familiar with Manfreda and the yucca pods, there are these chambered pods that are just stacked top to bottom with one seed after the next. You can get seriously like 100 seed from one good pod of a yucca or Manfreda. Like all those Asparagiaceae family plants, they all do this. They all have this survival strategy where they're like, hey, I'm gonna just produce all the seed I possibly can. And then I'm just gonna throw it everywhere. I'm gonna roll around for a year and disperse my seed all over the place. It's gonna tumble down the hillside, it's gonna land in between rocks. And if I'm lucky enough to get an ounce of water, I will germinate. So when you put these things in a tray for the first time, you just spread, you're like, I want a bunch of plants. You're gonna have way too many plants. I mean, the first time I did a flat of yucca, I did not know what to do with myself. I had hundreds of plants in a single tray. Separating them was work. That's the reason. It's a numbers game.
The research that you're working on. Tell us a little bit about.
Okay, so what is the actual research? So you got a little peek. This was something that interested me because there's not a lot of morphological, like I mentioned earlier, there's not a lot of like research done about morphological traits in early seed germination. So the plan is to at least do a 2-year study. And I'm assuming this is going to go 5 years, but in 2 years, we'll probably have enough information, enough data where we can say, okay, this is kind of what we're looking at. And we'll have enough case examples from enough different species, enough different genera that we can kind of pinpoint some interesting key factors. Morphology would be nice, but I'm not gonna bet on that horse. That's kind of not really, I don't think it's gonna be very dependable. So really what we're trying to see is, what's the time period between, germination to the cotyledon stage, from the cotyledon stage to true leaves, and then how long do they stay? We really wanna know kind of how long this lasts and how much it contributes to the overall survival strategy and early growth strategy of the plants. And I don't really think that that's been touched on a whole lot. And it would be really nice to do that on Texas plants especially, because that's. Jesse, 1, 2. Um, can I talk about some of the rare plants that were? Sure. Well, you saw the Salvia pensteminoides, the Penstemon triflorus, but we also have some of the rarest oaks in the world in our collection. Quercus hinklei, we also have, we have clones of that from the specimens that were planted in the garden very, very long time ago. We're very fortunate to have those trees. And we also have some new seed from the hinkleys that are barely struggling to survive out in West Texas. We also have one of the only collections of Quercus tornifolia, which is a red oak that was thought to be extinct, rediscovered in the Chisos Mountains. And we found a couple individuals there, and we were able to take some stem cuttings and graft them onto a wide range of other oaks, graft material, ranging anywhere from unknown, literally I had tags from the graft. The guy who did the grafts in South Carolina say unknown species. And then also some shoe-barred oaks that were grafted onto the Langtree oaks, just a lot of different stuff. That's a really cool one that we have in our collection. And then we also have some really rare legumes like Dahlia revertonii, which is blooming in the garden. Go check it out. And we also have wild collected Bauhinia, which is one you saw. Those plants, I guess you could call them cultivated since they were taken from plants in the garden, but the ones planted in the garden are wild collected seed. There's a lot of cool stuff. We also have, we're working on APGA collection for our milkweeds, so you can find Asclepias prostrata in the garden. Really happy to have that one. That was a 6-year endeavor from seed to planting. So that's a really cool one to check out. And we also have a bunch of really cool cactus like Astrophytum and I don't know, some really cool barrel cactus that we just recently got, Pharaoh cactus from an El Paso native plant rescue. So yeah, come to the garden, go to the rare endangered garden.
Smell bells, is that ever gonna be something that could be introduced, like a podcast back in '98 or something, talking about how it was in one area. I mean, I thought, well, what if, you know, the big turkey, I mean, there's no.
Yes.
There's only one.
Snowbells is one of those things that we really like to see back in cultivation, and we do have a lot of plants in the garden. And like you said, Paul Cox was a big factor in getting those established. So a lot of snowbells that we have that you can find them blooming right now, wow, ours are close to done. But I just actually found a population of styrax yesterday. I was driving around the hill country and I was driving down this nice steep canyon and I found 9 blooming plants, which is like a unicorn, guys. You found a population of styrax, are you kidding me? So this is a really tough one, and especially subspecies Youngiae, that's West Texas, and that's probably the rarest of all 3. And that's really tough to come by. Luckily, we have a good source on that, a cultivated source, but we don't have a good wild source. Do you guys know any people in West Texas who've got canyons on property? Hope you have. But yeah, that's something that we like to see in cultivation, and we're kind of cracking the myths behind that. Some people think that this plant is really difficult to germinate. It's really not. If you have styrax seed, collect it fresh, sow it fresh, leave it outside over winter, boom, in spring you'll have like almost 100% germination. It just really needs that very simplistic natural cycle. And don't store the seed, don't try and do any of that stuff. You're just tanking the germination rate. Each time you treat the seed, it's going down. I actually did a Pepsi challenge where I sowed fresh styrax, I scarified with a file, and I scarified with a knife. And the file did the worst. The knife was second, but the best germination came from untreated seed. Not stratified in a freezer or anything like that. It was just sown outside, let sit over winter, popped up in spring. So untreated seed is where I've had the most success with that. Yeah, I hope to see styrax in cultivation. We're working on it. I mean, we get some cultivated plants and some landscapes, 10 years, it's Styrax everywhere.
How successful is it in nature in terms of germination?
Oh, the Styrax? I,
Repeat the question.
so how successful is Styrax germination in situ in the wild? And I think that varies widely on conditions. I see it the most successful, like anytime you see a healthy Styrax population, it's always like a daredevil. It's on the edge of a 15-foot drop. You know, like if you can reach the speed, good for you. You're gonna have to climb for it. And sometimes you'll see them on slopes. So the prevailing thought is that the plant needs good drainage. It needs the right soil. They seem to prefer limestone more than anything else, but the West Texas subspecies is not that picky. So I would say they're very successful. I think a lot of stuff eats the seed. And I think they're playing the same kind of numbers game that everything else is. And it's like a lettuce tree, okay? If you have grazing deer in the area, they're gonna eat your Styrax. So I think a lot of them get munched down. They have too many extraneous factors to be very successful in the wild, especially when you look at the very sparse habitat they have left. Nothing can be successful in those places. They're very remote now.
I think Bamberger Ranch towards Junction City is still a program of trying to cultivate them and reintroduce.
Yeah, I hear Bamberger Ranch is doing work with Styrax. Actually, some of the seed that I tried to grow a couple of years ago was seed that had been stored. It was from Bamberger and it was part of that project and distributed seed. It was kept in dry storage and I had 0% germination on that. So that's one of the things that leads me to believe if we store this seed, we're setting ourselves up for failure. I hope I get more seed from that place. They have some cool plants.
Something like this. Is there a reason that you did the grafting rather than do a cutting?
Good question. With the tarfolia, it's a reason why we grafted instead of using other methods. And the reason why we grafted is because we could not get acorns from these trees. With oaks, you really don't want to try cuttings. They're highly unsuccessful with stem cuttings. There's some really cool research being done, especially in the UK, about doing stem cuttings from orchis, but we don't have enough material to just play like that. And I have tried a few stem cuttings with different plants, but we've been highly unsuccessful with that, which is expected across orchis. But the reason why we did grass was because stem cutting material, that big hardy stem cutting material, was the only thing that we could get access to potentially propagate them. I actually went back in late September, I think it was, after coming back from Arizona, we stopped in Big Bend, right? We live on the road, and we checked on the population, we were hoping to get some acorns, and it was, you gotta know, it was so dry. Oaks flower in the spring, and it was dry all spring. And they didn't get any rain until the monsoon finally hit. They had 0.0 inches of rain until July. So we went back that October and all the seed was aborted. We went to the tree, there's big, beautiful tardifolia, and the ground is covered in aborted acorns, just everywhere, like snow. And there's one pitiful, tiny little acorn hanging on the end of a branch. And that was all we saw. So we're hoping this year, Big Bend has had some good rain. There's a good year for rain for them. They had spring rains. If we get a good monsoon, who knows? We might get some acorns. Fingers crossed.
We've got a question on Zoom.
Hello, Zoom.
It's, are any of the plants you're studying going extinct themselves? And if so, does the work with cotyledons have any bearing on keeping these plants going?
So the question was, are any of the plants that we're currently propagating going extinct? And will the cotyledon research play into preserving those species? I don't know if it will or won't. The more we learn about plant propagation and every stage in the life cycle of vascular plants contributes to their survival in conservation. So I definitely think there's merit to the study, but whether or not it's gonna yield any results, it could just be a fun scientific experiment that helps us expand our knowledge, which is worth it in its own right. Many of the plants that we're working with are coming from the same type of plant. I think the Hinkley oak is, I know I talked about a lot, but we like orchids. The Hinkley oak is probably the best example of this because it's a relic species that is not fit to live in the climate it's currently in. This is not just a human factor, this is an environmental factor, this is a climatic factor that's impacting Hinkley oak. And it's a very small, petite white oak, it's very unique, it doesn't look like any other white oak I've ever seen. And it's very distinctive, and what we're seeing in the wild is it's beginning to hybridize with other white oaks in the area. And those hybrids are going to be the next generation of that offspring. Now, as far as the original Quercus hinkley genetic lineage, I think it's pining for the fjords, folks. I don't think it's long for this world. Enjoy it while you can. Come to the garden and see the ones that we have planted. There's 6. No, 7. There's 7 planted in the garden. So we're gonna keep it going. And we can clone these plants. They're rhizomatous, so we'll send up suckers. We can cut the suckers and root those. But as far as like genetic stagnation, yeah, I think we're kind of at the end. We're looking towards the future with this oak. We're looking at hybrids. There are a lot of species I work with that are going extinct. I would say most of them are going extinct because of habitat loss. That's definitely the biggest factor impacting our native wildlife.
I have a question about fresh seed. So I just collected seed on the plants that ripen, rather than save it and put it out in the fall. Are you saying I should put it out now and just?
100%. So the question was, since I brought up fresh seed, should you sow seed fresh if you just collected it? My answer is always going to be yes. This depends on person to person though, because you're going to have to do things how you can do them. Just like I have a lot of great resources to work with at the garden. I have a mist table, I have a grow space, I have greenhouses, I have tools at my disposal. I fight rats on a regular basis, semi-regular basis. But I have a lot of advantages with growing, and even I have to do things in line with the tools that I have. If you have the ability to sow fresh and protect that seed, like, I'm sorry, if you have buckeye seed, good luck keeping the squirrels away. They will kill for that fruit. In my opinion, it is always best to sow fresh. If you can, you should. If you can't though, there are some great storage methods. Most of those are going to be dry storage. Cold storage tends to induce dormancy, and you really can't predict how that's going to affect your crop. Dormancy has a lot of frustrating like barriers. It presents a lot of roadblocks. And with some seeds, like gumbumelia, right? Famous for everyone hating it in your garden, has a double seed coat and a dormant embryo. So if you manage to penetrate the 2 seed coats, the embryo itself has a period of dormancy. It is one of the most difficult plants I've ever read about to germinate. So that one, you can sow it fresh and you can wait 3 years. But the same thing applies to, what am I thinking of? There are a lot of trees that fall into this category. And hollies, ilex. All the species of ilex, you can sow a yaupon or a possum haw, and you can sit on that seed for 2 years. Viburnums, rusty blackhaw viburnum, everyone loves that plant. The seed takes forever to germinate. So it's one of those things where you really want to strike where the iron is hot, but you have to have a strategy to take care of your plants, obviously. It's gonna vary. So fresh, I'm gonna run on that platform. Anyone else?
If you, what is actually the purpose of the cotyledons? Do we know fully what that is, or are we still researching?
Yeah, so that's kind of an evolutionary holdover from the embryo. When you cut a premature seed open, normally you can kind of distinguish the parts. And there are a lot of really cool images of this online, right? Someone cutting open unripe seed, then the ripe seed, then you'll see different photos of the seed at different stages of germination. Oh, sorry, I didn't. Oh, dang it. Tom, what was your question?
What was the purpose of the cotyledons? Do we know? I was just typing it in.
I'm sorry, Sue.
I didn't
What is the
finish.
purpose of cotyledons in the life cycle of plants? What purpose do they serve? So definitely they're the first leaves to emerge. That's the plant's first opportunity to photosynthesize. Right? So it says, hey, I'm here, look at me world, I need some sugar. And obviously that's, you know, the first strategy. But as far as where they come from, this is variable because a lot of them are inside the embryo already. And some of them present as these weird little tissues that emerge above soil. So there is, I don't think there's a unifying factor in how they're observed, at least in the embryo. But this is definitely, I mean, it's defined as embryonic tissue. So I definitely think that this is an evolutionary holdover that functions as a way for the plant to make sure that there's all clear, you have oxygen, right? Because if you're gonna photosynthesize, clearly these things are photosynthesizing, they're green and chlorophyll. So what strategy, what do you need to grow? You're gonna need oxygen, right? So you can't be waterlogged, you can't be underwater, unless you're an aquatic plant, you want to be underwater sometimes when you germinate. But you can't be waterlogged, you can't be beneath the soil. And you have to make sure that there's enough light and oxygen to support your life cycle. And you can kind of observe this in plants that you grow in your backyard. If a plant wants more sun and you put it in shade, it's not going to grow like you see in situ. It's gonna present in some weird ways. The tissues will be stretched out and lean over and stretch for light. And sometimes will change colors. It will lose some of those distinguishing factors, like it'll become less or more hairy. It's just one of those case examples where you have to look and say, what do you want to do? And then it's gonna tell you. And I see a lot of, when I was photographing, I saw a lot of plants that just would, like some of the cotyledons would sit around for a long time. And I realized, oh my God, it's getting no sunlight. It's on a north, facing side of the greenhouse up against this wall at the edge of this pot. It's not doing anything. So maybe it's waiting for an opportunity. And these plants will exhaust their stored energy after they germinate. That's what those new leaves are for. So if they exhaust their stored energy, okay, well, you're on a clock. And if you can't get the nutrients you need, okay. I'm ranting. Is anyone else gonna ask a question?
Well, it's 18, so we probably should.
Okay, sorry, I'm gonna shut it now. Thanks, guys. It was great. Great to see you again.
There's a few plants still in the back of the room for those of you who aren't here.